Welcome to this video on how different types of microscopes work. Microscopes have revolutionized our understanding of life at the cellular level, allowing us to observe structures that were previously invisible to the human eye. So, from this invention of the first light microscope in the 17th century to today's advanced electron microscopes, these tools have helped us to explore the smallest unit of life, that is cells. In this video, we will cover the main types of microscopes, the light microscope, electron microscope, and then fluorescent and confocal microscope, examining how they work and their advantages and what they reveal about cells. All these will be covered in this video. Let's begin with the light microscope, the most commonly used microscope in biology. It works by passing visible light through the specimen and focusing the light with glass lenses. This allows us to magnify objects up to 1000 times the size. The light microscope is ideal for observing live cells and tissues. This makes it a fundamental tools in laboratories worldwide. The main components of light microscope includes the eyepiece, objective lenses, stage, and a light source. By adjusting the objective lens, we can change the magnification to view structures at different scales. The ability of observing living cells in real time is one of the major advantages of light microscopy, though its resolution is limited due to the wavelength of light, visible light, restricting how small we can see. This ability, that is observing cells which are live and in real time, is a key advantage of light microscope. This is not possible in electron microscope. We have learnt about light microscope in detail in the previous videos of this course, and also how to observe a slide under a light microscope and then how it functions, everything in detail in the previous video. Under a light microscope, we can easily observe the structure of onion cells, their cell walls, and even the nucleus. However, small details such as ribosomes remain invisible due to limited resolution. Now let's see an overview on electron microscope. Unlike light microscopes, electron microscopes use beams of electrons to image the specimen. This allows for much greater magnification and resolution. They can magnify objects up to 2 million times. This surpasses the limitation of light microscopes. However, they require the specimen to be prepared in a vacuum, so living cells cannot be observed directly under electron microscopes. They have to undergo preparation and a series of processes before being observed under an electron microscope. So, and there are two types of electron microscopes, the scanning electron microscope and transmission electron microscope. Each offers unique ways to view specimens. Now let's see scanning electron microscope. The scanning electron microscope, or SEM, is used to examine the surface of specimens. It works by scanning a focused beam of electrons across the surface of a specimen that has been coated in a thin layer of metal, such as gold or platinum. As the electrons interact with the surface, they produce signals that are converted into detailed three-dimensional images. SEM is commonly used to study the textures and structure of materials, including biological samples such as insect exoskeletons and then pollen grains and cell surfaces. One of the common use of SEM is to visualize the intricate surface structures of viruses or bacteria. These detailed images provide essential insights into how these organisms interact with their environment and then infect host cells. So this is the common use of scanning electron microscope. Now let's see transmission electron microscope, which is STEM. It offers a different perspective by allowing us to view the internal structure of cells. In TEM, a beam of electrons is transmitted through an ultra-thin section of the specimen. As the electrons pass through, they are scattered based on the density of the structures, creating an image on a screen. TEM can reveal intricate details of organelles such as mitochondria, chloroplast, and nucleus, providing us higher resolution than the light microscope. This allows us to see structures as small as few nanometers. Sorry. TEM is widely used in cell biology to study the organization of cellular membranes, the structure of organelles, and even the arrangement of protein complexes within the cell. These can be studied using transmission electron microscope. Now let's see fluorescent microscopy. Sorry. Fluorescent microscopy takes advantages of the ability of certain substances to emit light when excited by a specific wavelength. Fluorescent dyes are used to label specific molecules within the cells structures such as proteins, DNA, or organelles. When the specimen is exposed to a specific wavelength of light, these fluorescent molecules absorb the energy and then emit light of a different color, which can be captured to produce highly detailed images. This technique allows us to focus on specific parts of the cells in vibrant colors, making it a powerful tool for studying processes like protein localization, gene expression, and cell signaling in real time. That is, we can see that in live, unlike the electron microscope. And using fluorescent microscopy, scientists can track the movement of proteins within living cells, offering valuable insights into cellular functions and the progression of diseases like cancer. Now let's see confocal microscope. Confocal microscopy uses laser light and a technique called optical sectioning to produce high resolution. Three-dimensional images of thick specimens can be observed using confocal microscope. By focusing laser light on a very thin plane within the specimen and eliminating out of focus light, confocal microscopes create clear and sharp images of every layer of the sample. These layers can then be stacked together to form a detailed 3D representation. And confocal microscope is particularly useful for studying tissues, large cell structures, and complex cellular environments. In cell biology and developmental biology, confocal microscopes are used to image entire embryos or organs in 3 dimensions, allowing researchers to understand the spatial arrangement of cells during developmental stage. Now let's see the comparison of microscopes and their application. Each microscope has its own strength and is suitable for different types of research. Light microscopes are perfect for observing living cells in real time, but their resolution is limited, while electron microscope provide much higher resolution and magnification, allowing us to see the smallest cell structures, but they require special preparation and are not suitable for live specimens. Fluorescence and confocal microscopes, on the other hand, are specialized tools for visualizing specific cellular components in high detail and 3 dimensions. This makes them essential for molecular and cellular biology. In conclusion, microscopes are essential tools for unlocking the secrets of the microscopic world. Each type of microscope, from the light microscope to the electron and fluorescent microscopes, serve a unique function, and that is unique purpose, offering different perspective on cellular structures and processes. Understanding how they function and how they work allows us to appreciate the remarkable complexity of life on a microscopic scale. This drives scientists to discoveries and advancements in fields ranging in, sorry, from cell molecular biology and medicine to material science. Thank you for joining us in this exploration of how microscopes work. See you in the next video.